Good afternoon, friends. In last lecture, I discussed the biochemical test for bacteria, that is IMViC test. Today, we will see the same point, that is IMViC test. That first, the carbohydrate fermentation test, that is sugar fermentation test. This carbohydrate fermentation test is also called as what? It is also called as sugar fermentation test. What is the aim of this test, or why it is performed, or what is objective of this test? Yes, to determine the ability of microbes to ferment carbohydrates with the production of an acid and gases, or the production of acid or gas. Yes, what is aim of this test? To determine the ability of microbes to ferment carbohydrate with the production of acid and gas, or with the production of acid or gas. Next, we will see the principle of this test. The sugar is metabolized through the different metabolic pathways, and which is depend upon the type of microbial species and the aerobic and anaerobic environment. Yes, what I am saying, the sugar is metabolized is through the different metabolic pathways, and which is depend upon the type of microbial species and aerobic and anaerobic environment. If the fermenting bacteria are grown in a liquid culture medium containing the carbohydrates, if the fermenting bacteria are grown in a liquid culture medium containing the carbohydrates, they may produce the organic acid as a byproduct of the fermentation. Yes, what it produce? It may produce the organic acid as a byproducts of this fermentation, and these acids are released into the medium, and so the lower the pH of the medium. If the pH indicator such as phenol red, bromocresol blue is included in the medium, if the pH indicators, for example phenol red and bromocresol blue is included in the medium, the acid production will changes the medium from the original color to the yellow color. Then the gases are produced during the fermentation process, and it can be detected by use of small inverted tube, it called as Durham tubes. Yes, the gases are produced during the fermentation process, and it can be detected by use of small inverted tube, and this small inverted tube is called as Durham tube. Then within the liquid culture medium, if gas is produced, the liquid medium inside the Durham tube will be replaced by a gas in the form of bubbles. If the gas is produced, the liquid medium inside the Durham tube will be replaced by the gas in the form of bubbles. Then we will see the interpretation. If the medium changes from colorless to yellow and gas bubble is found in this tube, then it indicates the acid and also the production of gases. Yes, what I am saying, if the medium changes from colorless to yellow and the gas bubbles is found in this tube, then it indicates what? It indicates the acid and gas production. It indicates there is production of acid and gas, and in some cases the gas may not be evolved during the process. If no change is observed in the color of medium, then sugar is not degraded by the organism. Yes, what I am saying, I am repeating this. If the medium changes from colorless to yellow and gas bubbles, it is found that it indicates there is production of acid and gases, and in some cases the gas may not be evolved during the process. If no change is observed in the color of medium, then sugar is not degraded by the organism. If no change is observed in color of medium, then sugar is not degraded by the organism. Then the sugar is not degraded by this organism. Here we can see the observation of the tube. Nutrient broth plus respective sugar, it means it may be glucose, lactose, fructose. From left to right, it is uninoculated. Then we will see the first that is indole production test. In that first indole test, what is aim of this test? To determine the ability of microbes to degrade the amino acid tryptophan. Yes, what is the aim of or objective of this indole test? To determine the ability of microbes to degrade the amino acid tryptophan. Then we will see the principle of this test. Tryptophan, in presence of enzyme tryptophanase, it gives indole plus pyruvic acid and ammonia. Yes, in principle, the tryptophan, in presence of tryptophanase, it gives indole, pyruvic acid, it gives indole, then also it gives pyruvic acid, and it also gives ammonia. Then para-dimethyl aminobenzaldehyde plus indole, in presence of acid and alcohol, it undergo dehydration reaction. Yes, then the para-dimethyl aminobenzaldehyde plus indole, in presence of acid and alcohol, it undergoes which reaction? That is dehydration reaction, and it gives quinoidal red violet compound. It gives, there is formation of quinoidal red violet compound. Then we will see the interpretation or observation. The development of cherry red color at the interface of the reagent and the broth within the second after adding the covax reagent, it indicates the presence of indole, and the test is positive. When it is positive, if there is development of cherry red color after the addition of covax reagent, then it shows presence of indole, that is this test is positive. And if no color change is observed, that is there is no formation of cherry red color after the addition of covax reagent, then this test is negative, and so the organism are not capable of producing tryptophanase. Yes, what I am saying, what is observation? If the cherry red color is formed at the interface of this reagent and the broth within this few seconds after the addition of covax reagent, it indicates there is presence of indole. It means this test is positive, and if no change or if no color change is observed, then this test is negative, and so the organisms are, from that we can say that so the organism are not capable of producing tryptophanase. Here we can see the results. Tube on the left side, it means there is formation of cherry red color, that is E. coli. It shows present E. coli is, this test is positive for E. coli, and at right test tube it is negative. It means there is no formation of cherry red color after the addition of covax reagent. Here we can see the observation with images. Then we will see the next test, that is methyl red test. It is also called as MR test. Next, that is methyl red test. So what is aim of this test? To differentiate the E. coli, that is Escherichia coli, and E. aerogens, and to determine the ability of microbes to oxidize the glucose. To differentiate the E. coli and E. aerogen, and also to determine the ability of microbes to oxidize the glucose, and the ability of the microbes to oxidize the glucose with production and the stabilization of higher content of acid and product. Yes, what is the aim of this test? To differentiate the E. coli and E. aerogen, and to determine the ability of microbes to oxidize the glucose with production and stabilization of high content of acid and products. Next, we will see the principle of this test. That first, for E. coli, that first the glucose plus water, it gives lactic acid, acetic acid, and formic acid. Then plus methyl red, it gives carbon dioxide H2 plus red color, that is it gives positive taste and having pH 4. Then for E. aerogen, glucose plus H2O, it gives acetic acid, then it gives 2,3 butanediol plus carbon dioxide plus H2O and methyl red, then it gives yellow color and it shows presence of it shows this test is negative at which pH, that is pH 6. If there is formation of red color, then it shows this test is, it means the methyl red test is positive, and if it shows the yellow color, then it shows this test is negative. Then it shows this test is negative. At which pH, that is this red color is formed at pH 4 and this yellow color, it means it negative test, it is at pH 6. This is the methyl red test. There is use of MRVP broth, as I discussed in earlier lecture. What is MRVP broth? Then in this, the left test tube, it shows the positive, it means there is formation of red color, and at right, it means there is formation of red color, then this test is positive, and at the right side test tube, it shows yellow color, it means this test is negative. It means there is no any color change, or there is no formation of red color. It means this test is negative. Next we will see third test, that is August Prosker test, that is VP test. What is aim of this test? To differentiate the E. coli and E. erosion by the production of 2,3-butanediol and acetone via glucose fermentation. Yes, what is aim of this test? To differentiate the E. coli. To differentiate the E. coli and E. erosion by the production of 2,3-butanediol and acetone via glucose fermentation. Next, we will see the principle of this test. This test determines the capability of some organism. Yes, what this test? This test determines the capability of some organism to produce non-acidic or neutral end products. Non-acidic or the neutral end products such as acetyl methyl carbinol, that is acetone. Yes, what is acetone? That is acetyl methyl carbinol from organic acid that result from a glucose metabolism. Yes, what I am saying, this test determines the capability of some organism to produce non-acidic or neutral end products such as acetone, that is acetyl methyl carbinol, from what? From the organic acid, and that results from a glucose metabolism. And this test is characterized the E. erosion. The test identifies the bacteria that ferment the glucose. The test identifies the bacteria. The test identifies the bacteria that ferment the glucose and leading to 2,3-butanediol accumulation in the medium. We will see the reaction. The glucose plus oxygen, it gives 2-pyruvate, then carbon dioxide, alpha acetoacetate, then acetone and 2,3-butanediol. Next, the acetone plus alpha naphthol in presence of 40% KOH and absolute alcohol. Yes, acetone plus alpha naphthol in presence of 40% KOH and absolute alcohol, it gives diacetyl plus creatine, that is it gives pink color complex. So what is interpretation or what is observation? If the development of crimson red color, it indicates positive for E. erosion, and there is formation of, or there is no any color change, it gives negative test. Yes, what is observation or what is interpretation? If there is formation of crimson red color, then it indicates it is positive, or this test is positive for E. erosion, and if there is no any color change, and if there is no any color change, it shows it is negative taste. Here we can see this observation. MRB fibroth, there is formation of crimson red color, then it is positive. It means right left test tube, it shows this test is presence of red color. It means that this test is positive for E erosion. And at the right test tube or tube on the right side, there is no any color change. It means this test is negative. So this is the August Prosker test. Next we will see citrate utilization test. So what is aim of this test? Here the aim of this test is to determine the ability of microbes to ferment the citrate as a sole carbon source. To determine the ability of microbes to ferment the citrate as a sole carbon source. Next we will see the principle of this test. In this, the first, the citrate as a sole carbon source for their energy need. Then second, the presence of citrate permeases that facilitate the transport of citrate into the bacterium. Then sodium citrate as a carbon source and NH4 plus as a nitrogen source. Yes, what I am saying, the sodium citrate as a source of carbon, and then the NH4 plus as a source of nitrogen or the nitrogen source. Then the pH indicator is used in this test, that is bromothymol blue. Yes, what is pH indicator is used in this, that is bromothymol blue. Then this test is done on slant since oxygen is necessary for citrate utilization. This test is done on slant since or hence the oxygen, that is O2, is necessary for the citrate utilization. Next, when bacteria oxidize citrate, they remove it from the medium and liberate CO2. When the bacteria get oxidized citrate, they remove it from the medium and there is liberation of CO2. Then the CO2 is combined with sodium, which is supplied by sodium citrate. And this sodium which is supplied by sodium citrate and water. This CO2 combined with sodium and water to form a sodium carbonate, and it is a alkaline product. What I am saying, this CO2 is combined with sodium and which is supplied by sodium citrate and water to form a sodium carbonate. There is formation of sodium carbonate, which is an alkaline product. Then this raises the pH, turns the pH indicator to blue color and represent the positive citrate test. In the absence of color change, it shows the negative citrate test. It means if there is formation of blue color after the addition of this, then it shows this test is positive. If there is no any color change, then it shows this test is absent. Then we see the reaction. The sodium citrate in presence of citrate permeates or citrates, it gives pyruvic acid, oxaloacetic acid and CO2. Pyruvic acid, it gives what? The sodium citrate in presence of citrate permeates and citrates, it gives pyruvic acid plus oxaloacetic acid and CO2. Then excess sodium from sodium citrate plus CO2 plus H2O, it gives the sodium carbonate, that is it is an alkaline products. Here we can see the observation and interpretation. If there is formation of the color change, then it is, or we can say that this test is positive, and if there is no any color change, then we can say that this test is negative. Then we will see the nitrate reduction test. So what is aim of this test? The aim of this test is to determine the ability of some microbes to reduce nitrate to nitrite. So what is nitrate? It is NO3 minus. And what is nitrite? It is NO2. To determine the ability of some microbes to reduce the nitrate to nitrite or behind the nitrite stages. Then we will see the principle of this test. Certain organism like chemolithoautotropic bacteria, some certain organism like chemolithoautotropic bacteria and many chemoorganoheterotrophs can causes the nitrate as a terminal electron acceptor during the anaerobic respiration. Keep in mind what I am saying. Certain organism like chemolithoautotropic bacteria and the chemoorganoheterotrophs can use the nitrate as a terminal electron acceptor during the anaerobic respiration. And in this process the nitrate is reduced to nitrite, that is NO3 to NO2, by the enzyme that is nitrate reductase, and further reduce the nitrite to either the ammonium ion or the molecular nitrogen. Then the nitrate broth medium containing 0.5% of potassium nitrate, that is KNO3, then examined for the presence of gas and the nitriles ion and the nitrite ion in the medium. Here we can see the reaction in presence of enzyme nitrite reductase. Then sulfanic acid plus alpha naphthalene plus nitrite ions, these are the colorless, it gives the sulfa benzene, that is it gives the red color complex. If there is formation of red color, then it shows this test is positive. If there is no any color change, then it shows the absence of this test. Here we can see the images. First, if there is formation of red color, then it shows this test is positive, and if there is no any color change, then it shows this test is negative. Next we will see urease test. And so what is aim of this test? To determine the ability of microbes. Yes, what is aim of this test? To determine the ability of microbes to degrade the urea by urease. Next we will see the principle of this test. The urea is diamide carbonic acid. It is referred as a carbamide. Keep in mind, yes, what I am saying. Urea, it is diamide carbonic acid, and which is referred as what? It is referred as a carbamides. Then the hydrolysis of urea is catalyzed by specific enzyme, that urease. It gives the two molecules of ammonia. Here we can see the reaction. Urea plus water, it gives carbon dioxide, water and ammonia. Then urea attacks the nitrogen and carbon bond in urea. It forms the what? It forms ammonia. Then the presence of urease is detected when the organisms are growing in urea broth. Next, in this medium, the pH indicator is used, that is phenol red. Keep in mind which indicator is used, that is pH indicator, that is phenol red. Then splitting of urea create the alkaline condition, and which turns the phenol red to deep pink color, to deep pink in color. It is mainly used for identification of protease species from the genus of lactose non-fermenting enteric organisms. Here we can see the reaction. Urea plus water in presence of enzyme, that is urease, it gives carbon dioxide, water and ammonia. So what is observation or what is interpretation? If urea is present in the medium, then it will be degraded, which create the alkaline condition in the medium, which result in color change from reddish pink to deep pink. Yes, there is color change, that is reddish pink to deep pink, then it shows or it means this test is positive. And if there is no any color change, then we can say that this test is negative. Here we can see the observation for this urea test. Next we will see the TSI, that is triple sugar iron agar test. So what is aim of this test? To differentiate among and between the members of Enterobacterus family. So what is aim of this test? To determine the among and between the members of Enterobacterus family. So we will see the principle that first study and identify the organism belong to the Enterobacteriaceae family. Then it is used to distinguish this Enterobacteriaceae from other gram-negative intestinal bacilli. Then TSI agar slant, it contain what? 1% concentration of lactose and sucrose and 0.1% of glucose. Then in this test the pH indicator is used, that is phenol red. Then it is also incorporated into the medium to detect the acid production from the carbohydrate fermentation. Yes, this indicator is used or incorporated into this medium in order to detect the acid production from the carbohydrate fermentation. Then the uninoculated medium is red in color due to the presence of phenol red dye. This uninoculated medium, which is red in color due to the presence of what? Due to the presence of phenol red dye. Next, the yeast extract and beef extract, then peptone, it provides the nitrogen, sulfur, then traces of element and vitamin B complex. Then sodium chloride, it also maintains the osmotic equilibrium. Then lactose, sucrose and dextrose are the fermentable carbohydrates. Then sodium thiosulfate and ferrous sulfate makes the H2S indicator system, that is hydrogen sulfide. Yes, keep in mind the sodium thiosulfate and ferrous sulfate make the H2S indicator system. This thiosulfate is reduced to H2S by several species of bacteria, and H2S combines with and it forms an insoluble black precipitate, that is FeSO4, which is present in medium. And this blackening is usually occur in birth of tube. Then incubation is for 18 to 24 hours in order to detect, or what is the need of incubation, or why it is essential, because in order to detect the presence of sugar fermentation, then presence of gas production and H2S production. Why it is incubated? Because in order to detect the presence of sugar fermentation, then presence of gas production and the presence of H2S production. This is the need of incubation. Then the indicator is pink at alkaline pH, and the indicator which is yellow at acidic pH, and which is neutral pH is remain red. Yes, the indicator which is pink in color at what? At which pH? It is pink in color at alkaline pH. Then it is yellow in color at acidic pH. And it is red in color or remain red at a neutral pH. Yes, we will see the observation. In alkaline salt of the acidic bed, only glucose is fermented. Then acidic slant or acidic bed, there is fermentation of glucose, sucrose and lactose, that is all three sugars are fermented. Then bubbles or crack present, it shows there is production of gas or the gas production. And if there is formation of black precipitate or the present or if the black precipitate is present, it shows H2S production. I am repeating the observation. If alkaline salt or acidic bud, then it shows glucose is fermented. Then acidic slant or acidic bud, it shows presence of glucose, sucrose and lactose, all three sugars are fermented. Then bubbles or crack present, then it shows gas production. Then black precipitate is present, then it shows H2S production. Here we can see this test. Yes, I will stop here.